DNA Replication by Emily Furtado and Hannah Alton. For starters, DNA is a double-stranded molecule that is twisted into a helix, just like a spiral staircase. Each strand is comprised of a sugar-phosphate backbone and numerous nitrogenous base pairs, such as adenine and thymine as one base pair, and guanine and cytosine as another. Where and when does replication occur? DNA replication occurs during the synthesis phase of interphase in the cell cycle. The replication takes place in the nucleus. The first step in replication is to unwind or untwist the double helix by breaking the hydrogen bonds. This is accomplished by an enzyme called the helicase. The two untwisted DNA strands will now form a Y shape called a replication fork. Next step in the process. DNA replication begins at specific sites called origins of replication, where you see DNA polymerase. After the molecule splits longitudinally, forming the fork, DNA polymerase comes into play. DNA polymerase is the enzyme that performs the actual addition of nucleotides to the existing strand. Leading and lagging strands. Once the leading is added to the original strand continuously by DNA polymerase, the lagging strand, that is made discontinually by the Okazaki fragments, are linked together by the enzyme DNA ligase to produce a continuous strand. The nucleotides are assembled in the order that complements the order of the base pairs on the original strand to create base pairs. Half and half. Finally, hydrogen bonds form between the new base pairs, thus leaving two identical copies of the original DNA molecule. Though, when the DNA is replicated, two entirely new molecules are not created. Each new molecule has half of the original molecule. In summary, first, the helicase unwinds our double helix into two strands. The polymerase is added to the nucleotides to an existing strand. Ligase brings together the Okazaki fragments. Then the helix is cut and rejoined. Lastly, the RNA primase accelerates the synthesis of RNA primers.
We are now ready to review the steps of DNA replication. The process begins when the helicase enzyme unwinds the double helix to expose two single DNA strands and create two replication forks. DNA replication takes place simultaneously at each fork. The mechanism of replication is identical at each fork. Remember that the proteins involved in replication are clustered together and anchored in the cell. Thus, the replication proteins do not travel down the length of the DNA. Instead, the DNA helix is fed through a stationary replication factory, like film is fed through a projector. Single-strand binding proteins, or SSBs, coat the single DNA strands to prevent them from snapping back together. SSBs are easily displaced by DNA polymerase. The primase enzyme uses the original DNA sequence as a template to synthesize a short RNA primer. Primers are necessary because DNA polymerase can only extend a nucleotide chain, not start one. DNA polymerase begins to synthesize a new DNA strand by extending an RNA primer in the 5' to 3' direction. Each parental DNA strand is copied by one DNA polymerase. Remember, both template strands move through the replication factory in the same direction, and DNA polymerase can only synthesize DNA from the 5' end to the 3' end. Due to these two factors, one of the DNA strands must be made discontinuously in short pieces which are later joined together. As replication proceeds, RNase H recognizes RNA primers bound to DNA templates and removes the primers by hydrolyzing the RNA. DNA polymerase can then fill in the gap left by RNase H. The DNA replication process is completed when the ligase enzyme joins the short DNA pieces together into one continuous strand.
Bloopers. For starters, DNA is a double-stranded molecule that is twisted into a helix, just like a spiral staircase. Each strand is comprised of a